IPA's interaction networks help you understand how groups of dataset molecules might work together, whether by physically interacting as proteins or acting on each other via less direct mechanisms. These highly interconnected molecular networks are annotated with their likely biological functions, which can help you understand how they contribute to the biology occurring in your experiment. The networks are constructed to maximize their specific connectivity, which tries to find groups of molecules that are more connected to each other than to other molecules in the QIAGEN knowledge graph. The networks are then ranked in order of their significance with respect to finding that set of molecules in the given network by random chance. Here's an example of an interaction network, where you can see that some molecules act as hubs and some are less connected. The molecules in each network are listed here, and those highlighted in bold are the molecules that were eligible for the network from your dataset. These are also known as focus molecules, and are the analysis-ready molecules within your dataset. Those not in bold are either molecules in the dataset that were not analysis-ready, or those from the knowledge base that were added to maximize the number of connections between the focus molecules. The number of focus molecules is shown here, and the network score is shown in this column. This score is a negative exponent of the p-value calculated for the network by the right-tailed Fisher's exact test. The p-value is the probability of finding this set of focus molecules in the network compared to a set of randomly selected molecules. IPA identifies diseases and biological functions known to be affected by each network of molecules, and the top 3 categories are shown in this column. You can view more about the specific diseases and biological functions associated with the molecules in the network by selecting the Functions button. To view the network, click on the hyperlink for the ID. This will show you a new window showing a diagram of the relationships between molecules. The molecules that were added from IPA's knowledge base are shown in white, and dataset molecules that didn't pass your cutoffs or filters are shown in gray. The coloring of the focus molecule nodes is based on the data measurement values in your dataset. Networks can be merged by selecting the networks of interest in the table and choosing the Merge Network button. The new connections among molecules are shown with fuchsia lines. Networks that contain the same molecules can be viewed by selecting the Overlapping Networks tab. You can view the identity of these shared molecules by clicking on the relationship lines. For more information about networks results, you can go to the Help menu to access our online help manual or watch our video tutorials and webinars.